And lifespan.io. I'm Dr. Oliver Medvedik, coming to you from New York City at the Kambar Center for Biomedical Research at the Cooper Union, as always. And this week we have a special guest. We have Dr. Peter King introducing his work on a genome-wide association study, a really big one, looking at a variety of longevity and lifespan factors. And I'm just going to quickly share my screen here. And as always, in the background is Steve Hill and Fatima, who is helping organize all of this. And I'm going to just flip to the paper, which just recently came out. It's at elifesciences.org if you want to look it up. Research Communication, Genomics of 1 million parent lifespans implicates novel pathways in common diseases and distinguishes survival chances. So I've got a ton of questions here. I'm going to stop sharing. And this is basically, well, I mean, I'm going to basically turn the tables over to Dr. Joshi here. Am I pronouncing your name correct? Is that Joshi?
Peter Joshi.
Peter Joshi. I'm going to turn this over to Peter here. He's going to have full control of the controls for sharing the screen. And I believe you brought some slides to kind of help dissect this study, because it is a pretty complex study. You really did a meta-analysis, a lot of other studies, and you came out with a lot of interesting findings. You identified 11 novel, I think, genome-wide associations with lifespan. You replicated some others as well, and I wanted to kind of ask you a little bit more as you unpack all of this data from millions of samples. What are some of the surprises that you found, and kind of where, how this technology is going to be much more useful and usable as basically the databases get larger and larger, with our genotypes. This is clearly going to play a much bigger social role in a variety of medical backgrounds and insurance backgrounds and so on and so forth. So with that, I guess I'll turn the mic over to you.
Okay, thanks very much, Oliver. Okay, I'm going to run through some slides. Give me an indication whether I'm going too fast or too slow. There are about 60 slides in this deck. I'm certainly not going to present them all. Can you see the first one? It should just be a sort of cover slide. Genetics and genomics of human lifespan. No, can't see it. Okay. And now? Great. Perfect. Okay, well, thanks for that introduction, Oliver. My research is really about aging and the aging process. By background, I've done some actuarial work, and in more recent years, I've worked on human genomics. And so I've sort of brought those two things together here in a study of essentially a GWAS, as we heard already, of human lifespan. And we'll see whether or not I've managed into the aging process, having done that. Okay. Right, so what I'd say overall I'm going to talk to you about, and what the paper showed, was we're beginning to reveal how and how much DNA variation affects lifespan. We've actually constructed some lifespan predictions on the basis of what we're doing. And if we've got time and we're interested, we can maybe talk about some of the opportunities and challenges of the whole genomic revolution. Tell me if I'm wrong. I'm going to take it as read that people understand what a GWAS is and what DNA is. But I'll talk very quickly first about genetics, and genetics in the sense that Francis Galton would have understood over 100 years ago, and essentially familial resemblance. Galton noticed that tall parents have tall children, and developed something that he called the law of regression around that, which has led to what we now call linear regression and regression analysis more generally. Geneticists have interpreted that as a genetic variation within the population being passed down from parent to offspring. And sorry, my computer's pinging some other things that are annoying. I'm trying to turn it down. They've essentially assumed that that's genetic variation that's passing from generation to generation. So what we're really interested in, or I'm interested in the context of aging, is whether or not genetic variation that affects how long you live, i.e. is passed down from generation to generation. So the Galton way of doing this is really illustrated in this, and it's essentially saying lifespan does run in families. Here we're seeing mother age at death plotted against daughter age at death, and what we're seeing is that long-lived mothers have more long-lived daughters, but the X and Y axes are not on the same scale here. It's probably not entirely helpful. But the slope is not one-to-one, and in fact, the slope of this line is about 8%, which is to say that if your mother lives, I'll come to that in a moment, and that pattern is repeated across two different cohorts that we've constructed, and repeated from mothers to daughters, fathers to daughters, fathers to sons, and so on. So it does look like lifespan runs in families. As I was saying, the slope of these lines is about 8%, and possibly surprising to people. What I'm saying is that that if we look at a population and your mother has lived 10 years longer than average, the population of people that's true of will live only about 3/4 of a year longer than average. Okay, so those of you who may have parents that have lived to 90 and feel that you're bound to live to 90 too, it's far from the truth. You can expect an expectation of life of, if both of them live to 10 years longer than average, 1.5 years longer than your peers. Second thing to know is that actually the correlation between spousal lifespans is also about 3/4, or sorry, the slope is about 8% in that regression as well. So on the assumption that spouses tend not to share DNA, it's possible that the correlations we observe between parents, between parents and offspring, is a cultural or lifestyle correlation, not a DNA correlation. Obviously, those correlations between spouses and between parents and offspring are different, so I'm not suggesting we can just exactly adjust for that. But long-lived parents have slightly longer-lived offspring. That may be because of DNA, it may be because of lifestyle. But the effects aren't huge in either case. Do just interrupt as and when. I don't know to the extent that you're familiar with a geneticist concept called heritability, but it's essentially saying to what extent is a trait genetic, to what extent is a trait environmental. And here's some estimates of the heritability of lifespan. The old estimates were typically around 20 to 30%. More up-to-date estimates, my own estimate, which I'm saying is 16%, but might be less because my estimate's contaminated with environmental confounding. An estimate by Shor et al. Nearly 2 years ago now was also 16%, although I found it unconvincing that they had properly compensated for environmental confounding. And then much more recent and I think very robust study that was really nice last year was suggesting only 7%. So what we're saying there is 93% of the variation in lifespan seems to be due to, I think, mainly chance actually, but also lifestyle. And only 77% of it seems to be due to DNA. If you believe that estimate is the most robust one. I think we need a bit more work on this and other studies. The problem with all of these studies, as indicated in that last line, is you have to use some sort of model, and it's very difficult to prove the validity of the model. But anyway, it's
quick
small.
Question, since you did say I can interrupt. when you say heritability of lifespan, are we talking about average lifespan? Are we talking about maximal lifespan? Are we like what when you say there's a 26% or 7% heritability? What exactly does that mean as far as?
Okay, absolutely. So what I'm saying is that lifespan is a property that each of us has. In our case, is not yet expressed, but will have. You will live a certain number of years of life, whether it's 60, 70, 80, 90, 100. So you will have a number. I will have a number. And that varies across the population. And what I'm asking myself is, what's driving that variation? And what I'm saying is that essentially the variance, if you want to be very strict about it, the standard deviation squared, 93% of that, if we believe the bottom estimate, is driven by chance and the environment. And 7% of the variance is driven by DNA, because and the differences in DNA between us or across this population. So it's you're drawing your statistical distribution, and then you're sort of saying, right, how much of that spread in the distribution is due to what you inherited from your parents? Right. And I'm saying only a small amount.
And that's for the entire population. And somewhere, and I'm going to jump way ahead, but we can explore that later. That may be different if we look at individuals, take a subpopulation that are extremely long-lived, or versus the entire population of individuals. Is that correct?
So, I think that's I know what you're getting at. It's not quite the perspective of heritability. I think what you're asking is, if we take these particularly long-lived people, are they somehow more uniform, either in lifestyle or DNA? There has been suggestions as to the latter, and that the particularly long-lived have more genetic similarity than you would expect. But what we're here, as you're saying, Oliver, is I'm just considering the population as a whole.
We do have a question from the audience on Facebook. Jar Jar is asking, could the DNA stroke environment issue be solved using twin studies? Could we study enough twins to, would that help?
It's certainly one of the techniques that's been used. And you'll see here the historical estimates that we've put up were for twin studies. There wasn't a standard error in that first paper, unfortunately. So it's hard to know if actually it's really the same estimate as Jor et al.'s, and it's just a chance variation in the data or not. I am a bit skeptical about twin studies. I think there are twin studies, as I was hinting, along, and the same is true of all of these studies, are subject to having to use a model. And the question is whether that model is true, and essentially the similarity of monozygotic twins versus dizygotic twins follows certain patterns. I'm skeptical about them. This next slide brings out some of that skepticism. So the blue bars on this slide represent heritability estimates, predominantly using twin studies or family-based studies. And the orange bars reflect this study that was published last year that I thought was fantastic. I don't know if you or the audience are familiar with an Icelandic study called Decode. They've got some fantastic information in them. And they were coming up with much lower heritability estimates for just about everything than had been done through twin studies. And I find their method and model less prone to bias. So it's all data. You have to take any single study with a pinch of salt. You have to take any single method with a pinch of salt. But it may be interesting to people to understand that height does appear to be determined to a great part by your DNA, but geneticists actually don't know whether it's around 60% or around 80% DNA determined. whereas lifespan appears to be only slightly determined by your DNA, perhaps 7%. Hershkind and Magoo said 25%. Another recent study said 15%. So it's up for grabs across the field of genetics, not just the field of lifespan. But I think everybody is agreed that height is much more heritable than lifespan. Okay, so until now, I mean, I did say the words DNA a few times, but we haven't really talked in any real sense about DNA. We've talked about parents and offspring and the transmission and correlations amongst them. So the thing that I wanted to determine was, can we actually see a direct association between DNA and lifespan? And essentially, would somebody with genotype 1 perhaps have a survival curve that looked like the dark lines on this, depending whether they're a man or a woman, and somebody with genotype 2, i.e. different DNA, see a different survival? That's essentially what I'm trying to do. I'm dealing in survival curves because it's lifespan that we're studying, and that's at least I think the best way to think of a hazard model for survival. And in fact, as we'll come on to, that's what we did with Cox models. So this is essentially, we're really thinking of 1,000 people all starting here on each of these curves, and then if these people are all genetically similar, will their lifespan end up, or their pattern of survival end up like this, and so on and so on. Okay, I'll not go through SNPs in any great detail, or would you like me to talk through SNPs a little bit, Oliver?
Yes, a little bit. I mean, we're somewhat familiar, but
okay.
But please, yeah.
Okay. So the problem that I'm faced with is that part of your DNA might look like this. Your whole DNA would look like this, spread along 3 billion letters. Okay, so what on earth do I do with this? Well, part of the good news is that we now understand the function of this to some extent, and we could say this bit might relate to a certain gene. But then what does that gene do, and how does what it do affect lifespan? It's all very, very difficult. It's a code, essentially. And a code in the real sense of a code, it's not a metaphor. This code is translated into amino acids, which are then in turn translated into proteins. So how on earth do I figure out what this does? Well, the simplest way to do this would be to create 1,000 clones of me, and then 1,000 clones of me who were different from me at this particular letter here. And if we did that and then saw how long the first 1,000 lived and the second 1,000 lived and what killed them and so on, we'd have a very precise understanding of the mechanism of this DNA letter. You could imagine doing that, and I think they do it sometimes, reverse engineering computer code. You change a piece and you see what happens. Of course, that's incredibly unethical, impractical, etc., etc., etc. So we don't do that. But we're in the fortunate position that nature provides us with a natural experiment. If this is my DNA code, your DNA code might look like this. It would be the same nearly all the time, but occasionally it would differ. About 0.1% of the time it would differ. And then we could ask ourselves, well, how are those differences affecting how long we live? And the way we do that is actually by gathering many, many, many people who will differ in different ways at these different places. And we're then able to draw statistical inferences about the precise effect of this one single letter. Okay. These changes in DNA between you and I are called single nucleotide polymorphisms. A nucleotide is any one of these letters. Single means it's just one letter, and polymorphism means it changes. Okay, so a genome-wide association study is essentially scanning across the genome for these letters that vary between people, and there are about 10 million of them, and then seeing if each one of them individually associates with what we're interested in here, lifespan. So perhaps people who have this DNA letter live 0.1 years longer than this DNA letter if we get 200,000 of each of them. Okay, so this is a pictorial representation of the genome. If we microscope at your chromosomes, this is broadly what they look like. This is chromosome 1. They're not nicely ordered like this, I hasten to add, but this is chromosome 1, and so on and so on and so on. This is the X chromosome, and this is the Y chromosome. And so the essential question that we're seeking to ask is, does a change in the DNA at any particular point in this genome affect how long you live? That DNA at that point would be part of a specific gene. And the good news is that this has been done a huge amount over the last, I guess the Wellcome Trace Case Consortium study was 2007. So 12 years now. So for 12 years now, we've been doing this sort of study. And what we're seeing is thousands of associations between specific regions of the genome and specific changes in DNA and common diseases like digestive system diseases, cardiovascular diseases, and so on and so on. So the technique's working, it's robust, it's been replicated, all the great things of science. So genomic science has made massive progress and produced a whole lot of really good science, increased mechanistic understanding of disease. What do we know about lifespan in that way, in the way that I've just described? Well, in 2014, when I started this work, this is all we knew. We knew those thousands of associations between disease and the genome, but we only knew one point of the genome that affected how long we live. Some of you may have heard of this. It's the ApoE gene, and in particular, the ApoE4 variant was known to affect how long people lived. Nothing else. And this was essentially, despite really quite a large amount of effort having been put in to try to do what we described in the way that it had been done for disease, but essentially people had looked hard and found very little. And by looked hard, what they'd done here was, as we alluded to earlier, they'd gathered, I think, around 10,000 people who'd lived to beyond age 90 and compared them to people in more middle age and not been able to find any genetic variants that were associated with living beyond age 90, despite having 10,000 people who had done so. Okay, and this, again, maybe I will stop me if you feel I don't need to go over a Manhattan plot, but this is another pictorial representation of the genome. Instead of simulating what it looks down a microscope, we've got chromosomes across here, and every dot on this graph is one of those single nucleotide polymorphisms I talked about. There's about, I think, 3 million dots in this graph, and we're seeing whether or not any one of those DNA letters associating with lifespan. This is a 2014 study. And what you can see here is that there are the genetic variants in this region that are associated with lifespan, because this is a graph of the strength of the association in statistical terms. And what we're seeing is that there are no other statistically significant associations. This is the line for statistical significance in this experiment. And so that's what we saw in 2014. Sorry if I've dwelt on that a little, but it's important to have some perspective on that graph as we'll go through some more that look like this in a moment.
So can I interrupt for one moment? Just a quick question. Based on the picture that you showed, the slide, slide 29, with all those robust correlations that were done since 2007, genes associated with disease, maybe it's just a kind of a terminology, I guess, difference. So when you say genes associated with disease and only one, and in the next slide is, I think, ApoE4 on 30. When you say genes associated with lifespan, now all of these all of these mutations, all these SNPs that correlate with disease are going to obviously affect lifespan. So when you say there's only one associated with lifespan, you mean one that is actually correlated with higher than average lifespan at a certain age, correct?
Yes, or really what I'm saying is that I actually expect possibly many of these, if I'm going to be precise in my language now, many of the genetic variants associate that are highlighted in this graph probably associate with lifespan. But nobody had actually shown that association in any statistical robust sense. So I'm actually expecting a lot more than this, but only one had been proven in a study of lifespan directly.
Gotcha.
This is a sort of schematic of what was done in that study. We're taking cases and controls, and then we're testing the association between case control status and genetic variant status across the genome, one genetic variant at a time. Oops, going the wrong way. So instead of looking at people aged 90 plus, the sort of natural thing for me to do, coming from my actuarial world, is to actually look at how long people live. So rather than just categorize people as the problem with that is, particularly if you recruit a prospective cohort, you're going to need more people. It's better to study extremes than to study everybody. But you're also going to have to wait 50 years. We'll take all of our DNA today and then wait till we're dead, and then we'll know how long we lived. It's going to be 50 years from now. Not very practical. So the insight we had was to study parent age at death. If we take your DNA and ask you, what age did your mother and father die at? Admittedly, they may still be alive, but what age did they die at? And then test for an association between your DNA and how long your parents lived, it actually works very well statistically, because your DNA and your parents' DNA obviously have very precise properties, albeit probabilistic ones, across them. And so that works really quite well. And then what we did was actually slightly better than that. Because some of your parents will be alive, we said, well, actually, if your mum is 95 and alive, she's really interesting genetically too, or she's really interesting lifespan-wise too. She might be interesting genetically too. So let's take that information into account as well. So let's not just look at when parents died, but if they're alive, what age they are. For those of you who are familiar with Cox modeling, you'll sort of understand that now we're into the world of a survival model. And this is well understood, and we can do nice statistical association tests with the trait of, are you alive or dead? And if you're alive, what age are you? If you're dead, what age did you die at? And so, apologies if this slide is a bit busy, and I should mention Paul, the co-author on the paper that Oliver highlighted at the beginning, who did all of this work. I'm sorry he couldn't be with us tonight, but he's literally just back from a very long trip. So phenotype, the trait, lifespan, we're observing in the parents, are they alive or dead? And what age are they? And then the genotype, the DNA, we're measuring in the children ourselves. And then we're doing an association test. And for reasons we may get into a little bit more detail, we actually looked at 1 million parent lifespans. This was mainly enabled by UK Biobank, from whom we took about 350,000 subjects, 700,000 parents, and then we meta-analyzed that with 24 other cohorts around the world. So when you hear about big data and genomics, this is the sort of thing that people are talking about. We've got 1 million people in our study, and for each of those people, we have 10 million genetic reads. So I guess that's saying 10 billion different genotypes have been measured and then associated with 1 million lifespans. This is, broadly speaking, what the data looks like. We've got a distribution of dead fathers' ages at death, and then alive fathers' current ages. I won't go into Cox modeling unless I'm asked to, but it's a robust, relatively well understood way of testing the association between an outcome, the survival, and something like, say, BMI, or in our case, the genetic variation at a given gene. So I'll skip over this actually. This was the first study that we published, and it was great. As I say, there'd been lots of work done on this before, and in 2015, when this data was released by UK Biobank, we ran our analyses, and it was quite a nice result. Quite nice in the sense that we found this ApoE variant. So something seems to be working, because you're always worried with a new technique and a new set of data, is it going to work at all? So we found this ApoE variant did associate with lifespan, and with good, strong, statistically significant signal. And we found a second variant that associated with lifespan. A bit disappointing, we didn't find many more, of course, but it was interesting to note that this variant is very much about long-livedness. It's about late onset Alzheimer's disease. This variant, it turns out, is about smoking and nicotine consumption, and is about early death. But the key point being, in this context, we got some credible results out, one that was already known, and one that was, with the benefit of hindsight, not that surprising. So if we go back to those disease slides that we looked at earlier, this one would have appeared on the disease slide. Okay, so wind forward a couple of years, and this is the paper that we're really going to talk through now, the results of having understood the method. We've understood these Manhattan plots. I think for the purposes of today, let's not worry too much about the difference between the top and the bottom panel in this slide, but essentially it's to do with whether or not men and women have different effects of genetics on lifespan. And what we're seeing here is 12 genomic loci that are associating with lifespan spread out across the genome. We're seeing the two that we highlighted before, the nicotine locus and the Alzheimer's locus, and then quite a lot of new ones. So what are those? Because I suspect those gene names don't mean terribly much to you. Well, first thing I'd like you to look at in this slide really is the years. So this is how many extra years you would live if you carry the beneficial variant at this particular part of the genome. And typically it's only half a year, okay? So, and in the context of variation in lifespan, you know, the standard deviation of lifespan is about 10 years. So this is not immaterial, but on a personal level, if I tell you, oh, you can expect to live 0.3 years longer because you carry the beneficial variant at MAGE3, it doesn't mean a great deal, and so on and so on. A bit more of effect with this ApoE locus. And then second column I'd like you to look at quickly is the disease that, or the trait that this is known to associate with, and they're mainly cardiometabolic traits. So what we're saying is that in our study, with the statistical power we had, et cetera, what we found was variation in lifespan was driven by variation that affects cardiometabolic disease. And then finally, this frequency column, the nature of the way that we study things, we need quite a lot of people that carry one version of the genetic variant and quite a lot of people that carry the other copy. So this is basically common variation. As you can see, it's typically something like 40% of people have one variant and 60% of people have the other copy of the variant. We're not finding anything rare. You know, our sort of pipe dream is obviously to find some rare variant that adds 10 years to your life and develop some drug therapy on the back of that. We're not finding those, partly possibly because they don't exist, but partly because our study is not particularly well designed to find something that only 1 in a million people carry. Or sorry, even 1 in 10,000 people carry. we would struggle to find. I'll maybe, in the interest of time, skip over the next few slides. We then turned this all on its head, and going back to what I think Oliver asked about earlier, scientists know that disease affects genetic variation affects disease. So we said, well, let's go straight to all of those disease-causing variants and see whether or not they affect lifespan. And again, in the interest of time, I'll maybe just show the whole slide here and not try and confuse you too much with detail. And essentially what we found was we took about 30 variants, 30 of the strongest variants for Alzheimer's disease, smoking, cardiovascular disease, and cancer, and saw the extent to which they affected lifespan. And again, it's cardiovascular disease that's coming out. You can see the further you are away from this line, the more the variant is affecting lifespan. Positively here, negatively here. And you can see it's mainly cardiovascular disease we're looking at. To the extent we're seeing cancer-causing variants affecting lifespan, there are fewer of them. They're having less of an effect. And really interestingly, often actually a variant that seems to cause cancer is being associated with increased lifespan. This yellow box in the bottom right, that's a genetic variant that protects you against cancer, but actually shortens your life. That sounds bizarre. What's going on is that this genetic variant, while it's protective against cancer, is also causing cardiovascular disease. And so we've got a genetic variant that's affecting two things at once, or a genomic region, certainly, if we want to be precise, that's affecting two things at once in different directions. And so very much what I would say is we're seeing cardiovascular disease and smoking as the big things that are affecting lifespan from your DNA, or your DNA is affecting lifespan through those two pathways. We then really one of the principal purposes of genomic analysis is really then to begin to understand what's going on with cells, what's going on in biological pathways, which biological pathways might be intervening. And what we're seeing here is really quite a complicated technique, but what we're seeing is that actually, interestingly enough, the activity of variation that affects lifespan is particularly associated with enrichment and that activity in brain cells. So what I'm saying here is what's going on in your head appears to affect how long you live. A different sort of analysis, trying to do the same thing, but looking at pathways now. And what we see here is statistically significant things passing this dashed line. And what you can see is a preponderance of the yellow, and that's about lipid metabolism. So again, it looks like genetic variation for lifespan is giving rise to variation in blood lipid pathways, cardiometabolic pathways, heart disease, and then some, but less, in the brain again. Okay. So it looks it's about your heart and your head more than anything. We see some sex-specific effects and some age-specific effects. You'd not be that surprised. Some diseases men are more susceptible to and women are more susceptible to. So the next thing I wanted to go on and do is say, well, okay, so we've now identified genetic variation that affects lifespan. Can we actually make lifespan predictions? Could we at birth literally have taken any one of us, swabbed our cheek, sent it to any of the genetic testing companies and come up with a prediction of how long the person would live? And the short answer is, to some extent, yes. Okay. I don't know how familiar the audience will be with traditional genetic risk prediction, but traditional genetic risk prediction typically works like this. You would be in a family perhaps where breast cancer had been quite common. And if that was the case, you would go and see clinical genetics or a clinical geneticist, and they would say, yes, it does look like, based on the family history you've given me, that you could have a genetic variant that's strongly associating with risk of breast cancer. That's likely to be a very rare variant, so we'll test 10 different variants that we know are rare but really do affect breast cancer and see whether or not you carry one of those. And 99% of people, if we did this across the population, would be told you have the normal genotype, and maybe 0.1% even would be told that they would be carrying the genetic variant that was associated with higher cancer risk. And that would be very high. And for example, some of you will have heard that Angelina Jolie had genetic testing following the discovery of breast and ovarian cancer in her family, and discovered that she had a rare genetic variant that put her at very high risk of a breast cancer. And that's the sort of framework that typically happened. So we would start with a family where we had good reason to believe there was genetic variation affecting this risk, and then we would test one specific variant or maybe half a dozen specific variants and come up with an answer for these people. And often that answer would be that they did carry the variant, because that's why they had gone to visit the doctor, because of the existence of this in their family. What we want to do is something really quite different in one level. On another level, it's the same. We want to look at people's DNA and make predictions. But instead of looking at 1 or 2 points in the genome, we want to look at all of the information in this graph. So even these little spikes that are not deemed statistically significant from a genomicist gold standard point of view, there's information in there about how the genome affects how long you live. We're going to take all of that information. We're going to read your whole genome, combine all of that, and come up with a score for you and me and everybody else on the call for how long lifespan is. For your genetic propensity to a longer life. And for those of you familiar with statistics and the central limit theorem, because we're adding up so many things here, we tend to get a score that looks normally distributed, just like this on the right. This has been done for disease very successfully in a paper published by Amit Kira and Sek Thirasan about 6 months ago now, and they were saying, using that data I showed you for disease earlier, that they could predict susceptibility to disease in exactly the way that I described. And our question is, can we do this for lifespan? And the short answer is yes. By swabbing, let's assume that we're all of a similar age, by swabbing all of our cheeks at birth and running this score algorithm, we can make these predictions in the way that I've just described. And what I'm saying is that we're able to predict the bottom decile here living about 5 years less than the top decile. So if we were to take 100 of us, 10 of us would score the worst in the group and 10 of us would score the best. And if we then look at how long the whole of the 100 live, the 10 that scored the worst would live about 5 years less than the 10 that scored the best. For those of you who are into machine learning and artificial intelligence, we did use proper training and testing separation here, so there's not an issue of overfitting. We not only, this was done, sorry, the first graph that I'm showing you here was done in parents in the way that I've just described through this. We did the same tests in subjects. That works better. Sorry. Sorry, that bottom numbers. We did the same test in living people. We're able to do that in Estonia better than we can do it in our UK data, because we've got a wider range of ages, and we saw similar effects. We do admittedly see our scores are less predictive in Estonia than in the UK. That's possibly because the genetics of Estonians are different from British people, or possibly the environmental factors that affect their lifespan have been different. And that's worked across the different categories we've looked at. I think maybe just to sort of open out this discussion before I stop for more questions. The world is changing. Genomics is sort of at a threshold, I would say. As I've highlighted, genetic analysis and genetic prediction used to be about rare genetic variants that were specific to families. You used to see, or certainly in the UK, I don't know about in the US, you see screening during in vitro featualization for large effect familial disease. So essentially, a genetic test can be performed before the embryo is implanted for these rare genetic diseases. That's a framework in the UK. The sort of future we might be looking at is where, I think particularly in the US, it may be the case that people are just sequenced at birth because it's becoming sufficiently cheap. That would mean that risk scores for every trait imaginable could be constructed at birth. That could be height, it could be education level, it could be lifespan, it could be susceptibility to cardiovascular disease. Not saying that should happen, I hasten to add, I'm just saying that this is becoming technically possible. One of the problems is that you and I differ commonly at about 3 to 10 million parts of the genome, but each one of us probably carry 300 genetic variants that neither of our family carried. Neither of our parents carried, and will not be seen very commonly across the whole of the global population. And so variants like that, it's very difficult to tell what their effect will be. A psychological impact of having a risk score for Alzheimer's disease or lifespan or whatever, at the age of 18, if that might be when one might disclose it. And if you're interested in insurance, the insurer-customer information asymmetry that would arise if the insurer is not allowed access to that data. I've not talked about gene editing, but we've, I think, all heard about the Chinese researchers that recently produced gene-edited babies, or claim to at least, and we're seeing more and more gene therapies being trialed for major diseases. Okay, I'd just really like to thank all of the people that funded my work, the people that gave me the data, and the people that helped me do the work. And I'll stop there, and keen to take questions.
Great. That was a terrific introduction and description of the work that was done, Peter. Thank you very much for that. I actually have a question based on, I guess, potential interventions. So based on what you stated earlier regarding the range of heritability of lifespan, from a low of 7% from a recent study to perhaps as large as 25% or more. And you said the difference between the lowest and the highest decile of depending on the genetic variants an individual had, the range was about 5 years, correct? Something
right,
along that.
Yes.
So taking that together, is the implication of this that if you're in the lowest decile, that you can potentially increase your lifespan by about 93 or 75 to 93%, if that's the effect that the environment has versus the genome? Could you do some proactive interventions then, if the genome is playing that low of a role?
Yeah, I see where you're coming from.
Yeah.
I think the thing to realize is we're always talking about variation around the mean. So if we imagine the mean for the population we're discussing is perhaps 80, what we're saying is that I can predict some people will live 70 8 years, and some people will live 82 years across these deciles. And what you're saying is, well, yeah, and the environment is accounting for the rest of the variation, and that's 10 times that. And I think that's fair enough. We're really saying between 55 and 105 is the total environmental variation. You're then going on to, I think, make quite a strong inference that we could move everybody from the 55 to the, apart from the genetics, to the 105 by interventions. I'd be surprised at that. I think my gut feeling, but this is only gut feeling, is that more than half of what determines how long you live is just pure blind chance. The way that your arteries have sort of formed into a network around your heart, the way that your cells have divided, and if you haven't got a cancer and I have, that may have been due to radiation. It may just have been due to the DNA replication process. And I don't think we're going to have lifestyle interventions that will counter all of that, but certainly a very large part of it. And I mean, we see in the UK differences in lifespan between the top and bottom levels of society of 15 plus years. That's obviously about lifestyle, I would say. We've seen lifespan expanding across the past 4 decades by more than 15 years. That's about lifestyle. The genetics of our population hasn't changed material across those decades.
And kind of
sorry, that's a bit of a long-winded answer.
Yeah, that's okay. That's a good answer. And kind of a corollary to that is, so looking at the, and you said, you could potentially nudge yourself closer to the latter decile from the lower decile through interventions. Now, do you think that nudging would be higher for somebody in the lower decile pushing themselves to the higher decile versus somebody who's already in the higher decile and says, I want to live even longer, let me do some sort of intervention? Is there kind of a law of diminishing returns at that later stage?
I think directly looking at my work, I don't have a good answer to that question. I have seen some very interesting work about susceptibility to cardiovascular disease in Estonia. If what Krista Fischer, one of the co-authors on this paper, actually showed was that if you had a high genetic susceptibility to a cardiovascular disease, losing weight made a huge difference. Whereas if you had a relatively low susceptibility, it didn't make that much difference. So that would certainly accord with your hypothesis, Oliver, that actually it's those most susceptible that need to do most. And actually, if you're already in a good position, you can benefit yourself, but it won't have the same effect.
So there you go, Oliver, eat more salads.
Well, I don't know yet which decile I fall into, so maybe I should just keep doing what I'm doing. Just, not good things.
Oh dear. But I don't know, all the signs sort of seem to suggest that the environment and the lifestyle is the overriding factor, which is great news, really, when you think about it, because that is something that's malleable. That's something that you personally can, take control of, through better lifestyle, better, exercise and things like that. So great news for most people listening. You can do something towards that. It also suggests that epigenetics is probably more the dominant in terms of lifespan than the actual genome itself. We've got a chap in the chat called Flavius who's saying, I think epigenetic changes are what make you old.
What make you?
Yes, epigenetic changes, he says, and of course that epigenetic alterations are proposed as a cause of aging in hallmarks of aging. I mean, what are your thoughts on epigenetics in terms of lifespan determination?
So I think that's right. I'm not sure how familiar everybody is with epigenetics, but a epigenetic. So every cell in your body contains your complete DNA, and that DNA is, barring some nuances, identical across every cell in your body, and is what you inherited at birth. Epigenetics, however, is amongst other things, some marks around the backbone of the genome, and they control actually how the DNA behaves. And that then controls, for example, whether a cell becomes an eye cell or a nail cell or a bone cell or whatever. So epigenetics are fundamental to our biology. And as hinted, we're very aware that over your life, your epigenetic properties change, and every cell has different epigenetic properties. So aging and epigenetics and cell differentiation and development are all interlinked, absolutely. I guess where I don't disagree with the inference that it's been raised, but the question then is, what is causing these epigenetic changes? And what we know, for example, is that people with high BMI will see different epigenetic characteristics. So there's a sort of complicated biological pathway, and I'd say epigenetics is a fundamental part of that pathway. But can we analyze epigenetics as a thing in isolation, or is it part of a chain along that path? I'm less sure.
Yeah, I mean, there are a number of known contributors to epigenetic changes. You've got the microbiome in the gut, which is increasingly looking to be an influencer and also being influenced by the epigenome. So that's really exciting for me. And it's back to the eat more salads thing for us, I'm afraid, Oliver, because apparently the more fibrous materials you eat, the better the commensal bacteria, which means more short chain fatty acids, which is more butyrate and other things that help stabilize the genome. There's the telomeres, of course, as well. I wouldn't be complete without talking about them, of course. And they do have an influence over epigenetics via the telomere positioning effect. Now, there's an old chestnut from Wright and Shea, but it's still relevant. So, what the most exciting thing is about epigenetics is it's malleable, because we know that through full and also partial reprogramming with the Yamanaka factors and we know that Belmont and Ocampo, if you recall, that was their first journal club, I believe, Oliver, back in December 2016, they actually demonstrated epigenetic partial reprogramming in living mice. And I think that's rather exciting, given it may have a strong influence on lifespan. Could we rewind the clock? I think Steve Horvath would agree. So, any thoughts on that, anyone?
Yeah. Except we're still not quite sure why we all die at around 120, 125, and not 500. I mean, that's kind of getting to the heart of aging and longevity.
Well, yeah, I mean, there's a strong correlation between lifespan and genomic stability and epigenetic stability. I mean, look at creatures like the Greenland shark, for instance, that live, is it about 800 years? That's crazy. And they've got a super stable genome and epigenome. So there's definitely, there's definitely something in it, I think. And I don't think it's because they've been eating salads either in the case of the shark. So that's why I'm excited about partial reprogramming. So if we could get that done.
Well, I mean, it depends. I've been saying that all along. It depends on how you look at an individual versus a population, right? We are essentially a walking population of cells, and then the population of humans is, well, a population of individual metazoans. So in a very strict sense, right? I mean, the human species is essentially immortal. Somehow our germline has solved the trick of essentially resetting itself at zero continuously all the time that our somatic cells haven't, right? So there's mechanisms, fundamental mechanisms to be unearthed there as to what's taking place. But it seems like our collection of somatic cells, our lifespan that we have currently right now as an individual, conglomeration of 50 plus trillion cells, it seems that the environment plays a huge role in influencing that malleability.
That's all right. We can still repair it. There's still time for us. We can have that beach body. Well, I keep hoping anyway. I keep hoping. Let's hope the Salk Institute comes up with something real soon. I know that they're definitely moving steadily towards OSKM in humans, and I believe Dave Clare and George Church are also working on partial reprogramming, although I don't think they're using OSKM in that case. They're doing something else. But,
yes. So I think the practical take-home message is take care of yourself while you await these more radical interventions. Otherwise, you won't be around to see them.
Indeed.
Yeah, so we're back to the salads again, aren't we? Oh, God.
Those salads.
I don't like salad. All right, I'll have to. But,
yeah, I mean... You ask about, I'll see if there's a slide I skipped over, about phenotypic, trait correlations with lifespan. So this is something called genetic correlations that I'm looking at here between lifespan and various traits. Genetic correlations are quite interesting and quite good because a lot of the confounding that we see, so you're saying eat more salad. Are we confident that eating salad is good for you, or is it being of a certain socioeconomic stratum or whatever, all the classic problems with association studies? Genetic correlations don't suffer quite in the same way from that sort of confounding. They are still correlations, so I'm not saying this is causation, but might be of interest to you to see. These were the biggest genetic correlations we found between traits which had a known genetic basis. We tested, I think, probably 6 or 7 times as many traits and found these ones were statistically significant. So what we're saying is that a genetic propensity to be educated and a genetic propensity to live long are there, and that's the strongest effect that we can see. You might be more amused to know that there's a genetic propensity to be happy, and we see that genetic propensity to be happy actually correlates with lifespan as well. Sorry, the trait here strictly is death, and so actually you're seeing negatives for the good things and positives for the bad things. Before you get too smug, if you're feeling well educated, there's actually a negative genetic correlation between being happy and being educated, which is a bit of a shame. And you can see the other things here that you might sort of expect. We've got a lot of conventional diseases, all these cardiometabolic diseases, and then a genetic propensity to smoke in particular is bad for you.
So there you go. There's definitely that's definitely what your grandma would have told you anyway. Don't smoke. It's not a good idea. And as an ex-smoker of, well, about 20 years, I can tell you it's not a good idea.
Yeah, and actually, a lot of these interventions, everybody knows what they are. You don't have to have a professional tell you what they are. There was a study that was done. I have to pull it up and maybe show it to you, Peter. But it was a bit disheartening in that the amount of people who actually the percentage of people who were able to stick to a program or actually do something about an intervention that they knew was going to be positive or beneficial was abysmally low.
Yeah.
So it's not so much knowing what you need to do, but actually doing it, and that doing is really the problem I think most people have.
Oh yeah.
That's really it's not so much knowing that you have to eat this or knowing you're not supposed to smoke, but it's actually doing it. That the retention rate is abysmally low for a variety of reasons.
Yeah, I mean, we all know it, don't we?
We're sort of hopeful that, say, I talked about that cardiovascular disease susceptibility and BMI study. I guess we're sort of hopeful that, but through that personalized information and making it very clear to that particular group, that we might have more effect. But I think the jury is at best still out, or perhaps actually is in a negative about, even when you highlight that perhaps I'm at particular risk of cardiovascular disease, that even then it's hard to get me to stick to that regime.
Yeah. Can't count the amount of people I hear saying, oh, well, that's bad for you. You shouldn't do that. Yeah, but bacon, people. The thing is, people. It's back to that whole short-term versus long-term gains, and people. We're creatures of habit,
God help us if somebody comes up with a bacon-flavored cigarette.
Bacon-flavored cigarette. Yeah, you see that.
I shouldn't have said that.
Well, if you've just copyrighted it, you'll be all right.
I'm actually wondering whether there is actually one of those already, Oliver, because I know I recall many years ago in Italy there were different flavored cigarette tips. I'm not suggesting anybody goes out and smokes, but they had chocolate and caramel, and so I wouldn't rule out bacon flavored cigarettes. Be a bit. Ugh. But there we are.
Yes.
So yeah, but we all know the things. Don't drink to excess. Don't sit around and be too sedentary, because we know that there is a strong correlation with activity levels and longevity. All the usual things.
As we do sitting around all sedentary reading a paper.
And the irony is, the more I certainly found from my point of view, the irony is the more involved I've gotten with the field, the more sedentary I've become. How ironic is that? I write about longevity and aging research all the time, and ironically, I do it so much I don't do as much exercise as I should do. So, clearly, even the best of us are not without failings.
Well, I mean, with that, I think getting everybody healthier and happier is as an intervention should involve everybody out there, even the urban planners, right? Making a city, making the environment where you live in more conducive to living that sort of lifestyle. Me personally, this is just an aside, but I'm just going to toss this out there. I really hate going to the gym. Not so much that it's kind of painful, but more that it's boring. It is mind-numbingly for me boring to be on a treadmill and lift weights. But that being said, I have signed up for obstacle course races in the past, and I've run them, and I found them to be really stimulating and fun, even though they're exhausting. So, it's a matter of redesigning our environment, perhaps, in a way that would make us, live a healthier lifestyle, while at the same time making us happier. Maybe that's the trick to it.
Yeah, I'm just going to put a shout out
for
gardening.
And I think the same is true for diet. One of my colleagues studies the genomics of food preferences, and he shows, just a standard supermarket aisle. We were not programmed by evolution to cope with a supermarket. The amount of choice, the colors, everything there is just screaming at you, buy me, buy me, buy me. I think we need a different kind of world where it's easier to make good choices than the one we have at the moment.
Yeah, I mean, absolutely. Our ancestors were hunter-gatherers, and they would have often, stalked a gazelle for, dozens, even, or more miles before they made the kill, burning off a lot of calories, being very active, and also nomadic as well. We just don't have that in a modern lifestyle. And yet we have a body which is still geared up to escape tigers and hunt gazelles. And that's the problem. The solution, maybe get a big old rope and drag the trolley past really fast. So you have to chase it around the supermarket. I don't know. So some kind of interactive exercise when you go shopping. I don't know.
Well, perhaps
yeah.
next Journal Club we'll talk to some urban planners and I don't know, see how our environment can be improved for the better, and tap into our inner nomadic hunter-gatherer lifestyle.
Yeah, I can just imagine someone going through the supermarket and then looking for Pop-Tarts or something rubbish, and then a tiger chasing them down there. Oh, they wouldn't be expecting that, would they?
No.
Yeah, I don't think they would. Somehow, I think the health and safety would probably not approve that.
Well, if they survived, they'd be healthier. If they survived.
If they survived, yeah. But yeah.
Anyway.
The ultimate irony: we've got a caveman biology, and we're living in the future. So we've got to do something about that.
Anyway, well, thank you very much, Dr. Peter Joshi, for this wonderful explanation of the methods that you employed in this paper. We're certainly, as our databases grow, month after month, year after year, these techniques are going to be much more prevalent and utilized much more widely. And that was not only does your paper, have a role on studying these these associations on lifespan and longevity, which is what we here are personally interested in. But I think just the broader applicability of these techniques in genomes is going to be utilized much more widely, and that's just inevitable. So having more people introduced to these techniques and these methodologies, I think, is going to be beneficial for us all to understand how to use this data better. Thank you very much.
Thanks very much, Oliver.
And please thank your colleague and the rest of your colleagues on the paper, and your lead author Paul Timmers, who is recuperating from his long trip, and everybody in your lab and all of your associates. Thank you for this great work.
Will do. Okay. Thank you.
Yeah, thanks very much for joining us, and thank you to everybody that's joined us on the live stream. There are quite a few people today, and hopefully we've all learned something useful. I certainly have, and I'd also like to thank monthly patrons, the Lifespan Heroes, whose support allows us to make shows like this and all the other things that we do, and we do quite a bit. So if you enjoyed it, perhaps consider signing up and joining us as a Lifespan Hero. And you can find out more by visiting www.lifespan.io/hero. And we will see you next time with another exciting topic. Thanks very much, everybody.
Thank you.